Okay, good morning, everyone. Thank you for coming in. This is the first contributed talk session on decision-making and cognitive control. My name is Siddhant Iyer, and I'll be the chair introducing the speakers. So we'll get started with the first talk by Lucas Tian on a structured population code for a symbolic action grammar, alongside Daniel Hanuska, Kedar Gupta, Xiaoyi Liu, Xiaojing Wang, Josh Tannenbaum, and Winrich Freiwald.
All right, thanks a lot. I'm really honored and grateful to be chosen to give a talk. I'm going to talk about action grammar structure in primate frontal cortex. Imagine you're a gorilla, and your favorite snack is a nettle leaf. But the leaf has stinging parts, and you have to be clever about how you go about preparing this leaf to eat it. To deal with this problem, a troop of gorillas are known to deploy a specific action grammar, a set of hierarchical procedural rules for preparing these leaves. This action grammar seems to reflect structured symbolic programs. For example, the procedure to eat nettle leaves can be thought of as maybe a specific sequence of program calls. The procedure to strip stems may reflect a while loop to repeatedly perform the action until your goal is reached. Collecting leaf blades may similarly reflect a while loop. Symbolic programs analogous to these have been hypothesized to underlie diversity of human cognition, especially in abilities that involve generating novel thoughts and behaviors through rule-based recombination. This leads to the important point. Because action grammars don't require language and are potentially tractable in animal models, studying how they work may provide a powerful way to probe the neural basis of symbolic cognition. So far, however, we know little about how action grammars are implemented in the brain. And a major reason is that studies that have combined neural recordings and action sequencing have so far largely used two kinds of sequencing. Explicitly cued sequences involving specific ordering cues, like targets that you have to reach to in some order, or memory-based sequencing involving overtrained sequences. So we're lacking grammar-based tasks, specifically involving generative rules that are applied towards solving arbitrary novel problems. So to address this gap, we developed a grammar-based drawing task where subjects have to copy geometric figures like this. And the key idea is that ambiguous images like this probe grammatical structure. For example, one grammar involves repeating lollipops. Another grammar is to repeat all the lines, then all the circles. And another is to repeat all the circles, then all the lines. I'll tell you a bit about the macaque work in which we train them to learn and generalize these specific repeat grammars. Here's a video of a monkey performing the repeat grammar. He's generalizing this grammar to repeat circles and repeat lines. Although he gets to trace over the images, I want to emphasize that he gets no ordering cues at all, and this is a novel image. So I'll denote the repeat grammar in this manner, where A, B, and C are abstract roles to which specific shape strokes are assigned through learning, and N, M, and K are the number of repetitions which vary by image. Here's a specific example experiment. Trained on images like this, the monkey produces the correct behavior to repeat 2 lines, then 2 circles. To really see if he's learned the generalizable grammar, I test him on images not seen during training. And to be sure he isn't just memorizing solutions, I make these images harder. So they contain more image components, here's 6 instead of 4, and new spatial locations. And yet they readily generalize. So the monkey does 3 lines and 3 circles. So to quantify the behavior, I plot on the number of image components for a given shape on the x-axis, and the repeat length produced in behavior on the y-axis. And here I'm plotting actual behavior, taking only the first trial for each novel image. And you see the monkey demonstrates immediate generalization, including the harder problems. So we have a task where macaque monkeys learn and generalize repeat grammars. Now we can ask how grammars are encoded in the brain. So we implanted a large number of arrays to record simultaneously from 8 different areas of frontal cortex simultaneously. And in analyzing the activity, we found evidence for neural activity representing action grammars during drawing. And this representation was strongest in a specific area, the pre-supplementary motor area, or pre-SMA, which is an area on the medial wall of frontal cortex. So I'll talk about the 4 grammar properties that we found in neural data. I'll start with the representation of role. So it tested for activity that encodes the roles A, B, and C in a manner specific to the grammar task. So the monkeys switched between 2 different tasks on the same day, either using the grammar or a control single action task, in which the monkey produces a single stroke to draw images with only one shape component. And so it didn't have to use a grammar. This predicted population neural activity during drawing, where each ellipse represents activity during either roles A, B, or C. And it has separate states for each role, but only during the grammar task. As a contrast, the neural population does not encode the grammar, but instead encodes a specific shape or action, but have similar activity across these two tasks. So here I'm showing activity recorded in pre-SMA. It shows high separation during the grammar task and low separation during the single action task. So to quantify this, I devised a shape separation metric, which is the mean normalized Euclidean distance between shape clusters. As a contrast, consider activity in the ventral premotor cortex, or PMV. So this is an interesting area because we recently showed that it encodes discrete shape-specific action types, which we call action symbols. In PMV, activity was similar between two tasks, with high shape separation in both. To summarize this, I can plot the data this way, such that data points more to the bottom right reflect stronger encoding of role. So indeed, we see that pre-SMA is more to the bottom right compared to PMV. And here I'm plotting all the brain regions, and we see that pre-SMA has relatively strong encoding of role. So next, role abstraction. So we asked whether roles are represented in a manner that's abstract over specific shapes. So abstract role representations are useful because they support generalization over specific content, such as different shapes. For example, in language, the role of verb can be filled with many different possible words, like love or hate. And then complete sentences can be constructed by sequencing the roles of subject, verb, and object. And we could generalize this knowledge to understand and produce arbitrary new sentences like John loves Mary or Mary hates John. So I designed a task analogous to this situation here. The subject has to switch between two different shape sets with the same abstract ABC structure. And role abstraction predicts similar activity between shape sets. So here I'm showing an example of pre-SMA units activity during production of 3 shapes in set 1 and 3 shapes in set 2. And you can see that activity is very similar across these 2 shape sets, consistent with role abstraction. And as a contrast in PMV, many units strongly encode specific shapes, like this unit that encodes the green C shape. To quantify this, I use a similar approach as last time. On the x-axis, I'm quantifying shape separation within shape sets, and the y-axis, shape separation for a given role across shape sets. And in both monkeys, we see that pre-SMA has the strongest encoding of abstract role. So we next asked whether activity encodes ordinal position within each role. Alternatively, activity may not care about ordinal position. In the interest of time, I'll just tell you the result, which is that pre-SMA encodes ordinal position and does so more strongly than any other brain region. So come by my poster today for details. So lastly, I'll discuss the overall population geometry, and I'll do this by walking you through a summary schematic of the representations that we see in PMV and pre-SMA. So the pre-SMA geometry that I'm going to show you here is based on analyses that I don't have time to walk you through, but which again you can see in my poster. So consider the ABC grammar with 2 shape sets. I'll plot activity in mocked up neural state spaces. So first, during drawing of A, PMV encodes shapes as distinct categories, whereas pre-SMA encodes a role A in a manner that's abstract over shape, and further it has a linear encoding axis reflecting ordinal position. When you repeat B, pre-SMA has a separate orthogonal linear encoding axis for B, and similarly for C. So I'd like to highlight 3 implications of this and the related work that I mentioned. First, it provides evidence for anatomical separation of grammar structure and action content in PMV. A finding consistent or that speaks to long-standing hypotheses and models of cognition. Second, combined with a recent paper on action symbols, we're beginning to identify frontal cortical neural substrates that support grammar-based recombination. And third, our findings establish a foundation for studying how the brain generates novel representations. I'd like to thank my co-authors, as well as other people who have helped in this project, and funding sources. And also, I just want to shout out 2 posters today, one that's going right now with Carlos Correa, and another that I'm presenting later today. And thank you all for your time. Happy to take questions.
have time for one quick question. I have a question. So it seems like they can generalize to, if they're trained on maybe drawing 1 or 2 shapes, they can draw 5 or 6 of them. But do you think they could, any sense of whether they could compose the 2 shapes to make a third different shape?
Yeah, that's not something that they that's not something they spontaneously do. They learn each of the shapes as sort of their library of primitives, and then they'll reuse those in different sequences. Yeah.
All right, let's thank Lucas again.
Thanks.
Next speaker is Xiaoyi Liu, and the talk title is Orthogonal Task Representation Enables Flexible Task Switching in Human OPM-MEG and Neural Networks.
Okay, thank you for having me here. So in everyday life, we switch between tasks all the time. Imagine you're a barista, and you will need to switch between different mindsets to make those drinks here. And we can all agree that switching is hard. So how does a brain support flexible task switching? Traditionally, task switching is studied with a task where the relevant rule is only revealed at the beginning of each trial. So here, for example, the coffee orders. This approach treats task switching as a series of isolated events. However, in real life, a barista often gets a bunch of orders ahead of time, and they need to maintain and update those representations over a longer sequence of tasks. So how does a brain represent a sequence of tasks without them interfering with each other? To answer this question, we developed a paradigm where in each trial, participants would both see and hear a word. So here, for example, they would see left and hear right. And then they need to press one of the 2 buttons depending on the current rule. But instead of revealing the rule at the beginning of each trial, we organize the trials into mini blocks. And at the beginning of each mini block, we showed one of the 4 pairs of rules. So here, for example, we have read, listen. The participants then need to complete the first few trials based on the first rule. And then, after a generic switch queue, they completed the final trial based on the second rule. And importantly, the number of trials they need to complete before switching to the second rule is varied across many blocks, which means that our participants did not know when exactly the switch could occur. And in addition to humans, we also trained recurrent neural network on the same paradigm. And in this talk, I will start with the RNNs to test which internal representations might be helpful. I will then compare it to our human participants. So instead of training the networks to listen and read, we give them binary inputs in 5 channels. So here we have first and second rule, the visual and the auditory stimuli, and then the switch queue. We can then read the output of the networks as the left versus right response in human. And because we are most interested in what happens around the switch, we limited our main analysis to the last pre-switch trial and the post-trial. And in this presentation, I will use the different colors and shapes to denote the task pairs and the 2 trials. So here we can see that the networks learned the task quite well. And when the 2 rules were the same, we can see that the error rates were low in both trials. However, when the rule differed, the networks showed a cost not only after, but also before the switch, meaning that the upcoming task already influenced network behavior even before it became directly relevant. So what kind of internal representation are useful for this task switching? Here we predicted that to be able to perform the task, the network needs to encode the read-listen distinction along the task axis. So here, for example, this encoding axis distinguishes the pre-switch trial, with one end representing the read rule and the other end the listen rule. We then considered two representations the network might use to minimize interference. And according to a readout control hypothesis, the pre and post trials would use separate or even orthogonal representations for the corresponding tasks. And the RNN must control the readout to choose the correct task. Alternatively, the pre and post trial could use the same readout, and then the RNNs need to control the task representation to transform the task onto the shared task access when it becomes relevant. So to assess these representations, we extracted the hidden unit activity for the 4 task conditions in both pre and post trials. We then computed the cross-null bit distance between each pair of conditions and then visualized it with MDS. And now let's look at the pre-switch trial. Consistent with our prediction, the networks encoded the read-listen distinction of the pre-trial along the task axis. And to test the variability of this encoding axis across networks, we performed the encoding geometry analysis, which computed the cosine similarity of the encoding weights between runs or conditions. And this EGA confirmed that the RNNs did reliably encode the pre-task rule during the pre-trial. And in addition to the pre-rule access, we also observed an approximately orthogonal access encoding the upcoming task, even before it became behaviorally relevant. And this access is also reliable across our networks. So now let's look at the post-switch trials. And as before, we see that the same 2 access were preserved to reliably encode not only the current, but also the previous task. So how do the networks separate the 2 tasks? Using EGA, we found that within a given trial, the alignment between the 2 tasks is 0, meaning that the networks use an orthogonal representation to reduce interference. And this is consistent with a readout control hypothesis where the pre and post tasks use orthogonal representations and the RNNs need to dynamically change the readout to match the current context. So what about humans? To study this, we use OPM-MEG to measure brain activity while human participants perform the same task. And unlike the networks, humans showed a performance cost only after the switch. And the difference in behavior is also reflected in the internal representations. So as before, we computed the cross-null bit distance between conditions using the 192 MEG channels. And in the pre-switch trials, we observed a task access for the pre-task. However, unlike in RNNs, the post-task representation was much weaker. And similarly, in the post-trial, the brain only encoded the post-task, but not the pre-task. And importantly, the 2 task axes were reliably aligned with each other, suggesting that the same task access was reused when the task became relevant across pre and post trials. And this pattern is consistent with representation control hypothesis where the readout is fixed and the brain needs to transform each task representation onto that shared access when the task becomes relevant. So in summary, our results suggest that flexible task switching arises from different representational geometries in artificial networks and human brain. And what drives this difference and what are the downstream affordances will be an important direction for our future work. And we also have a poster this afternoon, so feel free to stop by if you have any question. And lastly, I would like to thank my bosses and our neuroimaging team, and of course my mentor for this project, Harrison Ritz. And thank you all for listening.
Okay, if there are any questions, feel free to come use the mic up front. But I guess you mentioned it's a future direction, so I don't know if you have any insight on what might make the RNNs have more human-like representations?
Yeah, there are actually, there are like a lot of findings that we didn't show here. For example, if you come to our poster, you will find that we actually find that during the earlier stage of the sequence, the RNNs are exhibiting similar representation as human beings. But what causes switch is still a question that we're studying. And there are also many different, for example, optimization constraints where the networks are only optimized for minimizing the errors. But for humans, we have limited resources, so we need to be energy efficient, and this could all lead to the difference that we observe in the representations. Thank you for the question.
All right, let's thank Xiaoyi again. Our next speaker is Theo Schäfer, who's going to present on dynamic belief state representations in human orbitofrontal cortex under uncertainty.
It's okay. Okay. Yeah, thanks for having me here. I'm Theo, and I'm very excited to present our work on belief states and their temporal dynamics in the human brain. Imagine you're following a football match on your phone via live updates instead of watching it directly on TV. It might be a match in which your favorite team will score a goal or one in which they don't. However, by just looking on your screen every few minutes, you cannot really tell which of these hidden states is true. One efficient way of dealing with the uncertainty of such partially observable environments is to form a probabilistic representation of the hidden state. In reinforcement learning terms, this is called a belief state, which is a posterior probability distribution over the hidden states given the available observations. Of course, these probabilities can be shaped by contextual factors such as the strength of your opponent, injuries, or a specific team lineup. But also the sheer passage of time can be informative about your belief state. It might make a big difference if what you think about this game if it's minute 12, 30, 50, or even the final minute of the game. So time-dependent belief trajectory for a positive outcome, let's say the first goal, could look like this. You start being optimistic, but after a while, your belief gradually shifts towards a non-goal or a scoreless game. Accordingly, if your team strikes a goal in an expected time window where the belief is high, we might expect a small reward prediction error. When the game is nearing its end and you almost lost all hope, but then a goal is seen in the final minute, then we might expect a higher prediction error. So time can be informative about your belief state if the environment has some temporal structure. It has been shown that the reward system, and particularly the dopaminergic reward prediction errors, are reflective of belief state-like computations, which has been linked to the prefrontal cortex. And in particular, the medial and orbitofrontal cortex are known to support hidden state inference and representation. So here we ask if we can track the belief state as it evolves over time in the orbitofrontal cortex, and if we can find related signatures in pupil, gaze, and behavioral responses. For that, we conducted an fMRI experiment combined with eye tracking. Here, participants acted as antiquities collectors, searching for items in different markets. There was a valued item giving 5 points, which we call the positive outcome, or O+, and there was a non-valued item, the O-, giving 0 points. Only one of these outcomes could occur in each trial, so there were rewarding and non-rewarding trials. And participants first saw a Q image, which signaled the start and type of the market. Then, after a variable waiting period, they saw one of the two outcomes and had to react to its location via button press. The two outcomes were separated on the screen on opposite diagonal positions so that they could measure anticipatory eye movements during the waiting period. The O- also appeared several seconds later than the O+, so that with increasing time, evidence was higher for a non-rewarding trial. Importantly, each trial belonged to one of three contexts. There was a certain deterministic A context with 100% reward probability, and 2 uncertain contexts, B and C, with 83 and 66%. We first looked at outcome response times as an index of how surprising an outcome in its timing was. Here we found an interacting effect of context and waiting time. So in the deterministic context A, participants got faster the longer they had waited for a reward, and in the 2 uncertain contexts, they got slower. This opposite pattern makes sense from a belief state perspective. When you know that reward is certain, every second that passes makes it more imminent, so reward prediction error shrinks. Whereas when reward is uncertain, then after a long time, your belief starts shifting towards a non-reward trial. So a late positive outcome is all the more surprising. This is why this behavioral pattern is well captured by a belief state reinforcement learning model and less so by control models that did not learn the temporal structure or treated the environment as fully observable. We were also interested if we can capture this pattern with a recurrent neural network that does not need to define the belief state representations, but learns them implicitly. And here we followed an approach by Hennig and colleagues in which a recurrent neural network did temporal difference learning on the task and got as input the observations as a time series. And then it gave a scalar value output for each time point. And this RNN reproduced nicely the belief state-like representations, and we could also decode belief state-like representations out of its hidden units. So even without hand coding these belief states, you can get similar representations out of an RNN. So next we ask if we can find these predicted dynamics in participants' eye data, so the gaze and pupil responses. And here, participants' pupil dilation responses to the positive outcome scaled with context uncertainty. So the more uncertain context, the higher the pupil response. And this interacted with waiting time. So the longer participants had waited for a reward, the smaller was the pupil response, and the opposite was true for the uncertain context. We also looked at participants' anticipatory gaze responses to the outcomes in the waiting period. And here we compared how close gaze was to the O+ versus the O- across the whole waiting period before the outcome appeared. And here you can see that participants initially looked at the O+ in all 3 contexts, and after some waiting time, they shifted towards the O- in the 2 probabilistic contexts, but stayed in the close to the O+ in the deterministic one. So they were faster, they were slower in the uncertain context to look at the positive outcome, and these switches between the O+ and the O- occurred in a substantial proportion of rewarding trials in the 2 uncertain contexts. Our behavior and eye data both suggest that beliefs about outcomes evolve over time. So then we ask whether the orbitofrontal cortex tracks this time-evolving belief. For that, we trained a decoder on a separate stimulus detection task in which participants were cued with an outcome category, for example, cat, and then they had to imagine this category during a waiting period, and afterwards judging if the provided image was an original or altered copy. We trained the decoder on the waiting period to predict different outcome categories, and then applied the trained decoder with OFC patterns on TR by TR on the main reward task in the waiting period. And this gave us a trajectory of decoding evidence for the different outcomes, here, for example, for the O+ for each context. And in the deterministic context, decoding evidence for the O+ first rose, and then remained relatively stable over the waiting period, whereas in the uncertain context B, it declined after a while. And this decline was more pronounced in the most uncertain C context, where O- evidence even exceeded O+ evidence after longer waiting times. So in sum, we find belief state-like signatures in behavior, gaze, and pupil responses, and in accordance with that, the OFC seems to track the belief state as it evolves over time. And with that, I want to thank you for listening, and my supervisor, Nico Schuck, as well as my collaborators, Andrei Sika and Tobias Donner, and the whole Latent Learning Lab in Hamburg. And if you're interested in further details, please meet me tomorrow at poster C16, session C. Thanks.
Thanks, Theo. If I understood correctly, the reaction times are going to be different for the 100% belief versus the other ones? Is that something that needs to be, or is that something that needs to be regressed out of any of the analyses, or?
Yeah, I think it's intertwined with the other measures. So I think we didn't do that yet. But I think it would be interesting to relate it. For example, if the decoding evidence for the O plus is higher in a certain context or a certain time point, then if the outcome appears, you should have a smaller reaction time, for example. So we didn't relate it yet on a trial-wise basis, but I think it would be interesting to do it. For example, there are, on a participant level, we found relationships between the pupil response and the OFC decoding. But yeah, we're still doing this on a trial-wise basis.
I see. Thank you. Thanks again. Yeah, okay. Okay, our next speaker is Zhuoyang Li, who's going to present on shared and distinct neural responses at event boundaries and task transitions.
Hi, everyone. Thanks for having me here. I'm Jill, and I'm very honored to present our recent work about shared neural responses at event boundaries and task transitions. Transitions are everywhere in our daily experiences. When we watch a movie, we segment the continuous visual stream into discrete scenes with boundaries between them. Over the course of a day, we switch between different tasks, for example, from making a coffee to having breakfast and then going to work. And in our own minds, our thoughts and conversations are also structured by this constant topic shifts. And in the realm of cognitive neuroscience, these transitions have been studied in two parallel lines of work, the event boundaries or event segmentation and task switching. In the event boundaries literature, people have used naturalistic stimuli such as movies to study the effect of event boundaries in memory organization. And in task switching literature, participants are often asked to switch between different classification rules. For instance, in this example, judging whether a digit is odd or even, or whether it is larger or smaller than a reference number. Despite the differences in paradigms used to study them, we think that both types of transitions have a lot of shared properties. For instance, they are both costly to process. We've seen this kind of reaction time effects in both event boundaries and task switching literature. And both of them are thought to involve an updating or reconfiguration process that helps reduce interference between different events or tasks. So given these commonalities between the two seemingly distinct phenomena, we ask what cognitive and neural processes are shared across different types of transitions. To answer this question, we took a deep sampling fMRI approach where we scanned 7 participants over multiple fMRI sessions while they performed a battery of tasks, including the task that we used to characterize event boundaries, which is a movie watching task, followed by a subsequent recall task. And for task transitions, we used 2 generation tasks where participants were given different prompts and asked to generate verbal responses. So for the movie watching task, participants watch a series of short movies, each about 5 minutes long, with boundaries between them marked by the onset of the next movie. After the movie watching session, they also did a movie recall task where they were asked to recall the content of each movie in whatever order they like. And we recorded and transcribed their speech and identified these recall boundaries at the end of each recalled movie. For the explanation generation task, participants were given short descriptions of specific scenarios and asked to generate as many possible explanations for that scenario as they can. Once they felt that they have exhausted all the explanations that they can come up with, they could say next, which will move them to the next scenario but, after a 15-second delay. And we defined each of the moments when they decided to switch as a task transition. Similarly, we had a word generation task, which is also known as semantic verbal fluency test, where participants were given these category labels and asked to generate words from the category. Similarly, we defined the task transitions as these moments when they queued to switch to the next task. So we first looked at the both activity at both event boundaries and task transitions. specifically the 15-second window post-boundary after accounting for the hemodynamic response delay. And we found that all types of boundaries evoked widespread activity increases in many different brain regions, most of which in the default mode network, including the posterior medial cortex, the angular gyrus, and the dorsal medial prefrontal cortex. So at the first glance, you might think that this just looks like a task-negative state, which makes sense but might be a little bit boring. So we also wanted to see if there's a structure to the neurostate on a finite scale. Therefore, we did a pattern similarity analysis by looking at the multivocal patterns in a given ROI at all types of boundaries, and then computed the similarity between these patterns, both within each condition and across different transition types. This analysis allowed us to ask, is there a generalized activation pattern between event boundaries and task boundaries? We focused on the core region of the default mode network, the posterior middle cortex, PMC, and as a contrast, we also included 2 ROIs in the prefrontal cortex and the ACC. So these are the cross-transition type similarity matrices for the 3 ROIs. First of all, in all 3 ROIs, we found a significant boundary pattern for each transition type. For instance, in this first cell, you can see that the boundary patterns at different movie offsets are significantly correlated with each other, suggesting that there is like a stereotyped boundary pattern for each type of the transitions. And looking at the off-diagonal cells, we found that only in the posterior medial cortex, the multivoxel patterns generalize across different types of transitions, but not in the DLPFC or ACC. And this suggests that there's a task-neutral state that the PMC resets to at different types of transitions. To better understand the properties of these transitions that produce this boundary pattern, we looked at the within-context boundaries. So for the movie watching task, we compared the between-movie boundaries that we've just looked at with the within-movie event boundaries, and found that although the between-movie boundaries are quite similar to each other, they don't look like the within-movie event boundaries. And this is consistent with prior work by Li and Chen using the same movie stimuli. And similarly, in the explanation generation task, we found that the between-task transitions have a shared activity pattern, but is distinct from the within-task transitions. So now I've hopefully conveyed to you that this generalized pattern exists in the PMC across different types of tasks. But what does it mean, or like what does it tell us about the underlying neural processes? So we looked at the individual subjects' responses in the PMC at each transition type. And you can see that there are these subregions in the PMC that are more active at the boundaries than others. And we also know that there are these interdigitated networks within the DMC that can be measured by functional connectivity. So we took this approach and computed the connectivity of each voxel in the PMC to the DNA and DNB subnetworks, and found that in all subjects, the regions that are more active at the event boundaries and task boundaries are also the regions that are more connected to the DNA subnetwork. So we think that this suggests that there might be a resetting or like a context updating process that is mostly dominant by the DNA subnetwork at different types of boundaries. And this could serve to clear out the current situation representation to reduce interference for the next incoming event or task. So to summarize, we found broad activity increases at both event boundaries and task transitions, especially in the default mode network. Beyond univariate activity increase, we also showed that the multifocal patterns in the posterior medial cortex generalize across different event boundaries and task transitions, and didn't find that in the DLPFC or ACC. Furthermore, we show that the generalized boundary pattern is linked to the DNA subnetwork, and we hypothesize that the more anterior parts of the PMC activations are likely related to cognitive control, given their connectivity to regions like the DACC. With that, I'd like to thank my advisors, Chris Honey and Janice Chen, and other lab members. If you want to follow up with questions or feedbacks, please feel free to reach out, and also come to my poster tomorrow at C15. Thank you so much.
Any questions from the audience?
Hey, yeah, over on the other side. Really cool talk. It seemed like there was some structure in PMC where like word generation and movie watching, let's say, were more similar to each other. I don't know if like,
Here?
Yes, so it seems like they're in the far left. It seems like there might be some structure there. I don't know if those upper triangular blocks are significantly different from each other, but I'm wondering if you might speculate about that. I mean, it seems like one thing is that movie watching and word generation might be easier than movie recall or explanation generation, but I'm wondering what other kinds of structures you think might be in
yeah, that's a good question. So first of all, like those cells, they were not significantly positive, so we're not sure if there's like a kind of similar extent to which like the boundaries patterns generalize. But we think that there are like a lot of commonalities between different tasks. For instance, the movie recall and explanation generation tasks both kind of involve activating different schemas and like transition from each other at boundaries. So we think maybe the shared pattern might have to do a little bit with that, but we're not sure kind of entirely how. Thank you.
Okay, let's thank the speaker again. Our next speaker is Daniel Deng, who's going to talk about model-based error signals in the frontal cortex, precede successful inference and hippocampal context updating during human latent-state inference.
Awesome. Thanks so much for having me. Today I'll be talking about model-based error signals in the frontal cortex and how they can help us support human inference. So what exactly is inference? Well, here I use it specifically to refer to the process of detecting changing environments and adapting one's behavior to match that. With us living in this highly complicated world of ours, it's pretty simple to see how that could be a very crucial computation just for our day-to-day processing, right? And really a major entry point into this process is by simply observing the outcomes as we interact with that environment. For example, imagine you're walking down the street to your favorite cafe only to find a dead rat on your burger, right? And without needing to try the food, really, you'll probably decide on the spot that this place is no longer worth coming back to. To illustrate this process even more explicitly, let's consider a scenario where you learn to press the right button in response to a monkey image and the left button to a car image. Now, the very next time you see a monkey, you were expected to press the right button and get it correct. But in fact, the opposite was true this time, and that should really get you thinking. So it turns out a latent response inversion has just occurred, and if you have that structural understanding, you should really be able to rapidly adapt your behavior policy to all subsequent images just based on that singular incorrect feedback. And the key insight here is really that the brain needs to do two things. It needs to first have a context representation, which concerns the structures governing the environment, as well as perform outcome interpretation, right? So what does the feedback actually imply? And when you see an error, does it reflect a genuine mistake or signal true changes to the underlying process? Now, the example I'm showing is exactly the setup for this 4-stimuli reversal learning task we've used to investigate the inference process, for which a response inversion, or as we call it, a context change, occurs covertly every tens of trials. We recorded this task with neurosurgical patients implanted with depth electrodes for their epilepsy monitoring. This actually allowed us access to record both unit spiking activity as well as local field potential during the task performance in both the frontal cortex and the hippocampus. Now, a previous study from the lab actually already concluded that the hippocampus actually carries a context variable, but only when the subjects are capable of doing inference, meaning the ability to adjust responses were yet to be validated stimuli since a context change. And this already indicates to us that there's a relationship between representation and the inference behavior. But this understanding is global, and we don't really know the details still around the timing inference. In particular, we don't really know how the feedback itself is going to be used for the inference, nor do we know if the frontal cortex, right, regions we know to be associated with adaptive behavior and error monitoring is going to be involved. On the other hand, we also don't know how the hippocampus is going to be dynamically updating its own context representation at the time of inference. So to better understand the behavior and to facilitate the downstream neural analysis, we designed for this task a two-stage reinforcement learning model. The model carries two variables, B and Q, respectively for belief over context and expected value under fixed context mapping. Action will then be selected based on the belief-weighted Q values. And here on the right, I'm going to show the actual update rules governing this model, as well as some example traces for how B and Q would actually move. But in the interest of time, I won't go into the actual details. To summarize concisely, the model first observes outcomes and then uses that to update the current belief, then update the value only if the outcomes are not sufficiently explainable by the belief mismatch. Now, by assessing the model accuracies on trials requiring inference, we can actually distinguish sessions by their ability to perform inference. In one group, we have sessions where subjects basically get things immediately correct after a switch and after a single error. We dub them IP for inference present. And then there were those that lack such ability. We call them IA for inference absent. And to quickly define some nomenclature, we label trials with yet to be seen stimuli since a switch with integer positions, 1, 2, 3, 4, and those that are in between with range notations, for example, 1-2. Trials after all stimuli have been visited are going to be called steady state or SS. Now, we also specifically denote position 1 as the switch trial and position 2 as the inference trial. In fact, those trials are the ones that emit the most interesting behavior correlates, with switch trials having the most surprising outcomes and the inference trial having the most uncertain responses. And by the way, this becomes important soon. We operationalize outcomes with unsigned prediction error or surprisal greater than 0.5 as being surprising. With that, we can finally look at some neural results. Decoding from pooled pseudopopulation neurons, we found that the frontal cortex actually carries very salient error signal regardless of one's inference capability. But that signal persists until post-feedback period, or error integration, as Theo will call it, only on the IP sessions. Now, this result is confounded by the surprisal content of the errors. So next step, we asked if the frontal cortex can actually distinguish between the error types, and if that explains the persistence. As a baseline, we found no meaningful differences between the surprising and unsurprising errors in IP sessions. But for IA sessions. But for IP sessions, that distinction is actually salient, with anterior cingulate tracking the surprising error immediately during feedback, and pre-SMA tracking them thereafter. Unfortunately, we don't have enough to decode in VMPFC, but one can infer that such model-based signal exists there too, and can help explain the persistence. And here are some examples showing exactly this model-based tuning to the outcome surprisal. Now, it turns out to us that the error integration is actually not limited just to that brief window of the switch trial, but is actually available all the way until the end and validation of the inference trial. Specifically, we found anterior cingulate again significantly reactivates on the inference trial around the time of response and persisting all the way into the feedback period. Note that this is not an error signal as we analyze only correct trials. In fact, we compared the neurosignature of anterior cingulate between back-to-back recorded sessions. We've actually found that the strength of the signal is directly correlated with the same person's ability to perform inference. So clearly, it seems that the frontal cortex, especially anterior cingulate in this case, encodes the evidence for inference until confirmation throughout a period where inference is actually being carried out actively behaviorally. Now, to complete the puzzle, we must also look at how the hippocampus encodes its context relative to behavior and this inference signal. So in the steady state, context is actually encoded in both IP and IA. But only in IP do you see a clear pattern generalization to earlier trial positions. In fact, this is going to be staircase such that on and before inference trial, the context remains strongly in the pre-switch configuration before switching to the correct one. And this is confirmed if we look at alignment of the context coding directions across all possible trial positions, where you see clearly opposing clusters sort of delineated by the end of the inference trial. And again, here are some example units showing this rapid reorganization. And note that the polarity is going to be inverted for the lighter traces pre and post inference. Now, finally, as a work in progress, we actually found that these context-tuned hippocampal units selectively phase lock only to anterior cingulate theta rhythm during the inference trial, showing potentially a direct link between outcome evaluation, inference processing, and context updating. So all in all, the hippocampal representation is going to be lagging both behavior and inference, perhaps in a dependent manner, rapidly updating only thereafter to the correct confirmation. So in summary, we used a simple RL model to identify sessions first by their inference abilities, as well as distinguish between the two types of errors of interest. Using that, we found that the frontal cortex actually carries model-based signals that signal whether or not inference has been triggered, thereby putting them perhaps at the center of the inference processing. On the other hand, hippocampus, while encoding a context representation, seems to only update afterwards, suggesting that the hippocampus alone is insufficient for the inference process, and perhaps it only plays an indirect role, or must be used in conjunction with the frontal cortex. Now, as the future goes, more work is definitely required to understand the causal interactions between these brain areas, as well as between this inference and context signal we're so interested about. And with that, I would like to thank my co-authors and all of you for your attention, and I'm happy to take any questions.
I have a question. So you split the sessions by inference present versus absent, and you found it only in the present?
Only in the present, yeah.
So in the inference absent sessions, are they picking up at all the switch behaviorally, or not really?
It's a mixed bag. Some people have, perhaps, some sessions you have a late realization, so it perhaps takes more than one surprising errors. It will take like maybe 2 to 3 unique positions of incorrectness to make you realize, oh, something has changed, and they might adapt correctly for the fourth one. But the more common strategy is that they actually just get confused. So, and that's actually something that you see in monkey work too, is that once you have this highly surprising event that perhaps does signal to them something about the world is changing, but they don't really know what the correct structure is, they just perform some sort of resetting or gets confused in their response policies such that everything hereafter is going to be chance level. Yeah, but still, even then, they don't exhibit any sort of neurosignature for that switch or the trigger in their brain.
Cool. Thank you. Thanks again.
Thank you.
Okay. Our final speaker is Akhmetzhanova, who's going to talk about the title being From Impulsivity to Engagement. Latent behavioral states reveal distinct decision strategies in rats.
Sounds good. Hi, everyone. Thanks for joining. My name is Zaya, and I'm excited to present my work on how latent behavioral states can reveal distinct decision strategies in rats. So probably this was you at the start of the session, sitting up straight, listening carefully, and fully engaged. But as we're getting closer to the end of the session, you might find yourself drifting, checking the clock, and thinking about food. We can call those two latent states engaged and disengaged. You might remain in the engaged state for a while before transitioning to the disengaged state, although I hope my talk will bring you back to the engaged state. In our work, we want to understand those internal states, specifically how model-derived latent states can produce behavioral signatures that go beyond the model's outputs and drive state-dependent neural dynamics. And to study this, we use a free response change point detection task. So the task begins with the rat positioned in the center port, ready to begin a trial. Then the rat hears a sequence of pulse-generated clicks for some variable wait time. At a randomized time in the trial, the click rate increases, and the rat has to report the change by moving from the central port to the side port in order to receive the water reward. And there's 3 possible outcomes for each trial. So a hit occurs when the rat correctly identified that there was a change in the click rate by moving from the central port to the side port. Then a miss occurs when the rat failed to detect the change by staying in the central port. And a false alarm occurs if the rat moved to the side port before there was any change in the click rate. Our dataset includes 6 rodents tracking across 57 distinct behavioral sessions, and the design of this task overall aims to evaluate how rodents track and accumulate evidence over a short time scale to guide their decisions. Then to infer the latent states, we use the generalized linear model hidden Markov model, so the GLM-HMM. Since the GLM part involves the direct input to output mapping, our input feature includes stimulus trains, which is defined as the change in the click rate. And then the input metrics will be defined for every trial and have a value for the stimulus trains and an offset, which is going to be a column of ones. Our model architecture involves end states, which could vary from like 1 to 5 or whatever you input, wherever you pass it. The model also knows about the 3 categories representing the 3 possible outcomes for each trial. We also want our states to have standard transitions, meaning that no external input will affect how they transition back and forth, but the observations will be impacted by the stimulus strains, by our input. And finally, our output matrix will include the behavioral choice that a rodent made on every trial. So the matrix will be defined for every trial and have a categorical value for the choice. So overall, given our input feature, we want the model to fit our output data of the behavioral choices and spit out end states that could describe how each category is represented within each state. And so what we find is that the cross-fault validation of the JLM-HMM model revealed that 2 states were sufficient to capture the overall decision-making behavior in this task. As you can see here in this inferred state sequence plot, the 2 states remain stable over blocks of trials or even entire sessions, with transitions occurring intermittently rather than every trial. Next, we wanted to characterize those states by calculating the distribution of each outcome within each state. So here in the first state, we see a high predominant rate of the false alarms, so we call that state impulsive, while state 2 showed a pretty much reduced false alarm rate and more correct detection, so we call that state engaged. More importantly, in the impulsive state, the hit probability did not depend on the stimulus strains, while in the engaged state, the correct detections were shown in the stimulus-dependent manner. What was surprising is that those behavioral states produce the signatures that go beyond the model's output of our rodent's choices. For example, we saw that evidence accumulation window is actually longer in the engaged state. So here in the behavior reverse correlation plot, we show the click rate preceding the false alarm response. And in the false alarm engaged trials, the integration window of the stimulus evidence was actually broader, so it was about 300 milliseconds before the response. While in the impulsive false alarms, the integration window was much shorter. It was about 250 milliseconds before response. Another behavioral signature was shown in the response timing. So the impulsive state had much shorter response time, both for the false alarms, which were aligned to the stimulus onset, and then the heat trials, which were aligned to the stimulus change point. We also saw that the reaction times of the hit trials were actually dependent on the stimulus strains, which is what we expect for the correct detections in a stimulus-dependent manner. And finally, we found that the engaged state had increased the word port engagement, characterized by a higher number of the side pokes, longer individual side poke durations, also longer cumulative time in the side port, as well as faster reentry intervals. So together, these results show that impulsive and engaged latent states predict important aspects of behavior that go well beyond the model's outputs, capturing how animals integrate evidence, time their responses, and seek reward. To probe the neural basis of our latent states, we recorded the high-density electrophysiological recordings using the NeuroPixels probe. So we targeted 2 regions in the key decision-making network. So it's the frontal-oriented field FOF and anterior dorsal striatum. So with more than 100 recording sites, NeuroPixels probe recorded hundreds of units per session, far exceeding traditional techniques. And what we saw so far was that the mean firing rate at the stimulus onset was pretty identical between impulsive and engaged trials. We also tested that across other different time periods, and we found no significant signature between the states. That suggests that individual neurons' firing rate alone could not distinguish between the impulsive versus engaged state. But using the populational pattern, we could decode states on a trial-by-trial basis with pretty high accuracy, shown here in the yellow curve. We also saw the same result across the rodents and across the different sessions. But here's the catch. So the decoder trained on the time elapsed, early versus late, could decode time on a trial-by-trial basis with very high accuracy as well. So that creates a potential complication that we're currently addressing in our research. Is the population code truly representing underlying behavioral state, or are we simply decoding other related factors such as passage of time? And if you have any ideas or suggestions about this, I would be happy to talk to you after this talk or at my poster presentation tomorrow. And just to bring everything together, the JLM-HMM model revealed that 2 latent states optimally capture behavior in this task. One of them was impulsive state with frequent false alarms and behavior largely independent of the stimulus strains, while another state we called engaged with reduced false alarm rates and more correct detections in the stimulus-dependent manner. So the states also mapped onto direct observable behavioral signatures such as evidence accumulation window, reaction time, and reward port engagement, extending well beyond our model predictions of a rodent choice. And so while the populational firing pattern could decode states on a trial-by-trial basis with very high accuracy, near-perfect decoding also leaves an open question of whether neurorepresentation reflects the true underlying state or other related factors such as time. And I just want to thank my advisors, Dr. Tim Hanks and Dr. Rishi Chattery, and all the members of the Chattery and Hanks Labs. And if you have time and you want to learn more, come say hi to me at my poster tomorrow at 2 p.m. Thank you so much.
Any questions from the audience? Okay, I have one. So it seems you're able to decode the two states, and these states are comprised of stuff like faster versus slower reaction times, false alarms, etc.
Mm-hmm.
Is it possible to make sure that the decoder is not actually, just differentiating between reaction times as opposed to the entire state of being engaged versus impulsive?
Yeah, so the decoder that we trained actually did not know anything about reaction times. It only knew that, oh, this trial was impulsive, this trial was engaged, so it had no information about the reaction times. The reaction times is something that we saw as a behavioral signature that we did not pass into our model. So the Zhuoyang model that I was talking about was actually fitted to the choice that Ronald made. It knew nothing about the reaction times, but when we found those two states, it actually showed the differences in the reaction time, which was, I think, pretty cool. So it goes beyond the model's outputs. And the decoder here only knew about the population of pattern and the state label. So we did not pass anything about the reaction times, but maybe that would be something that we could consider.
All right, cool.
Thank you.
Yeah, let's thank all the speakers once again, and thanks everyone for coming.